Good morning friends, good morning students. In today's class I will discuss reaction of monosaccharides, right? And in this video I will discuss all these reactions. First of all I will start with tautomerization and anodization reaction. Then I will cover the reducing properties, then oxidation reaction, then reduction reactions, then dehydration, and then last but not least Osazone formation, and finally I will discuss the formation of esters, right? So first of all I will start with the tautomerization and anodization reactions. Now I will start with the definition of tautomerization. The process of shifting of hydrogen atom from one carbon to another results in the formation of indiols, and this reaction is known as tautomerization, right? And the sugar possessing anomeric carbon atom undergoes this reaction, and moreover, let's take the example of glucose, right? So, for example, we have glucose, and we would kept it in alkaline solution for several hours, right? And in alkaline medium, right, when glucose is kept in alkaline medium, it will undergo isomerization to form d-fructose and d-mannose, right? And this reaction of conversion of glucose to other intermediates like fructose and mannose is known as Lobre de Bruyn von Eckenstein transformation, right? And this reaction will result in the formation of our indiols, right? And these indiols are very highly reactive in alkaline medium, right? And these indiols are intermediate, right? So let's discuss the example. We have glucose here, we have mannose here, and we have fructose here, right? We are shifting hydrogen atom from one carbon to another, right? So glucose, first carbon aldehyde group, HOH, and over third carbon OH this side, right? And in mannose, first carbon OH and second carbon, third carbon OH this side, right? Mannose. And fructose, we have keto group, right? So we are shifting a hydrogen atom from one carbon to another, which will result in the formation of common intermediate that is known as indiol, right? So anomeric carbon atom containing sugar kept in alkaline solution, right, for several hours will result in common intermediate that is known as indiol, and these reactions are known as tautomerization reaction, right? Now I'll start with the second reaction. Now I will start with reducing properties, right? The sugar can be categorized into two categories. First is it is reducing sugar, and secondly it is non reducing sugar, right? And moreover, the reducing property is attributed to the anomeric carbon free aldehyde or free aldehyde or keto group of anomeric carbon atom, right? The sugar is reducing when it has free aldehyde or keto group at anomeric carbon atom, right? And it is non-reducing when it doesn't have any free aldehyde or keto group at anomeric carbon atom, right? Several tests are in part for that for using this property, right? First is our most favorite Benedict's test, right? Second is our Barfoot's test, and third is our Felling's test, right? These tests are attributed to the reducing properties of sugar. Now I'll start with the third reactions. Now I will start with oxidation reactions, right? Depending on the oxidizing agent used, whether the first carbon is oxidized or the terminal alcohol group is oxidized, the reactions products are different, right? For example, when the first carbon, first group that is aldehyde is oxidized, we will get our gluconic acid, right? But if we oxidize at terminal alcohol group, what we will get, the product is different, that is our glucuronic acid, right? So oxidation depends on the oxidizing agent used and which carbon atom is being targeted. If the first carbon that is containing aldehyde group like in glucose, it will be converted to gluconic acid. And if terminal alcohol group is targeted, we will get our glucuronic acid, right? And this group can, in case of fructose, we will have keto group here, right? So that covers with our oxidation reactions. Now we'll start with the next one. Now in reduction reactions, the aldehyde keto group is converted to its corresponding alcohol by strong reducing agents like sodium amalgam, right? So 2H positive are added and it is converted to into its corresponding alcohol, right? So let's discuss some examples of common monosaccharide like glucose. Glucose is converted to D-sorbitol. Galactose is converted to D-dulcitol. Mannose is converted to D-mannitol, right? Fructose is converted to D-sorbitol plus D-mannitol, right? And this ribose is converted to D-ribitol, right? And these have some clinical implications as well. For example, this D-sorbitol, right? It can accumulate in tissue and also cause increase in osmotic pressure, right? For example, if it accumulates in the lens, right, of the eye, right, it will cause increasing osmotic pressure and it will lead to the complications of cataract as well, right? And moreover, it can also lead to peripheral neuropathy and nephropathy as well. In that issue, it will cause increasing osmotic pressure and will lead to the complications as well, right? And there is one other compound that is our mannitol, right? It causes it is used in decreasing the uh, intracranial pressure, right? Because it causes loss of excess water from the body, right? Now I'll start with the next property. Now starting with dehydration properties, right? We have a monosaccharide glucose here, right? When it is dehydrated with concentrated sulfuric acid, the three molecules of water are eliminated, right? One from here, one from here, and one from here, right? That will result in the formation of our hydroxymethyl furfural, right? Meanwhile, if we have pentose, it is converted to furfural, right? If hexose is there, it is converted to hydroxymethyl furfural. If pentose is there, it is converted to furfural, right? And these compounds, both, they condense with phenolic compounds like alpha naphthol, right? And they will form colored complex, right? And this forms the chemical basis of our Molish test, right? So Molish test is based on this dehydration property, right? And if we have some polysaccharide or oligosaccharides, right, they are first hydrolyzed to corresponding monosaccharide and then they are further dehydrated with concentrated sulfuric acid to form furfuras, right? So that finishes with our dehydration reactions. Now I'll start with Osazone formation. Now in Osazone formation, the phenylhydrazine with acetic acid, right? Acetic acid is CH3COOH with glucose, right? Will result in formation of glucosazone, that is our Osazone, right? When you see these crystals under microscope, they have particular shape, right? For example, if we have glucose, fructose, and mannose, we'll get needle shaped crystals. And in case of maltose, we'll get sunflower shaped crystals. And in case of lactose, we'll get powder puff, right? And if monosaccharides, which are different at C1 and C2, right? But since phenylhydrazine binds here, so the difference of carbon 1 and 2 is unmasked, right? So if the sugar, they are different as C1 and C2, they will give the same type of Osazone, right? But the difference will not be masked if the carbons are 3, 4, 5 or 6, right? But C1, C2 is masked by phenylhydrazine because it binds there, right? So if some sugars like monosaccharide, like we have mannose here and glucose, the carbon 1 and carbon 2, the difference is unmasked by phenylhydrazine. So the crystals are same data, needle shaped, but in case of maltose, will get sunflower, lactose will get powder puff, right? We'll start with the last property, that is formation of esters. Now the last reaction, that is our formation of esters, right? So alcoholic group of monosaccharides, right, can be esterified, right? And these can be done by enzymatic reaction as well as non-enzymatic reaction, right? And esterification of carbohydrate is the most common example of ester formation, right? For example, we have glucose, it is phosphorylated, like glucose is here, ATP donates the phosphate moiety and it can be converted to glucose 6 phosphate or glucose 1 phosphate. This is very good example of ester formation because ATP donates the phosphate moiety to form ester, right? We'll get glucose 6 phosphate when glucose is phosphorylated to form glucose 6 phosphate to glucose 1 phosphate. These reactions are involved in glycolysis and some other metabolic pathways of carbohydrates, right? So that's this is with our complete lecture on reaction of monosaccharides. For upcoming videos, please like the channel, subscribe it, and press the bell button below, right? So you will get notified when I will upload my new video, right? Thank you. Keep sharing, keep studying. God bless you. Bye bye. Thank you.